Good morning, everybody. It's time to get started. Time to get started. Thank you. Welcome to day 2 of Caos. We're going to have 2 speakers this morning. Then we'll have our coffee break. And this afternoon, I just want to remind people that we have the award-winning abstracts. And that means there's actually going to be talks. Some people were apparently confused that we were just going to hand out medals or something. But no, they will give talks, so you all need to make sure you're here. All right, so promptly after lunch. Actually, just all listen to Sharon. She'll whip us into shape. Okay, so we're going to get started, and keeping with a long Caos tradition, there are no introductions other than say that our first speaker is Bevil Conway, and there's his title right there.
All right, thank you. Great. All right, so I'm going to try and stay on time here, see if that works. Okay, so I first want to thank the organizers, and especially Marius and for doing all of that emailing and getting me here, and Alex for his support over the years I've been at NIH and his friendship, and to the rest of your team. It's been really fun meeting many of you who I don't know this community very well, and of course it's one that my work is sort of gravitating towards. So before I launch in, I just want to acknowledge the team of people in my group that's done a lot of the work, and I'll try and mention them as I go along. But it's been really fun working with this group of people. Okay, so much of my lab's work is motivated by these 2 questions. What is color for, and how does the brain do it? The motivation here is obviously because I'm interested in color, but it's more because I think a fuller understanding of those questions can be a useful tool for understanding how the mind and brain work. So my central argument here is that, color is cool and interesting and fascinating, but maybe we can use this as a tractable system in the same way people have been using events and so on, as a tractable system to gain an understanding of the full sweep from sense data up to cognition and behavior. So one clue to the importance of color vision in high-level behavior is amongst mammals, excellent color vision has evolved only in certain non-human primates, macaque monkeys being one of them and us being the others. And of course, we're also endowed, like macaque monkeys, with a big brain, and we have an even bigger frontal cortex. So why has it been so difficult then to pin down the role of color vision, what color is for? So if I have one goal today, it's to persuade you that color is not just a low-level stimulus feature. I know a lot of you probably think, there are great papers out there arguing that color serves these high-level functions, and yet somehow still in the way we interact with each other, even in this conference yesterday, you heard talks where people said, look, I'm going to just change the color of the ball. This is just a low-level feature. I'm just going to make, that new thing. And I'm only picking on Anna because it was a nice example, where we sort of reflexively treat color as this low-level stimulus feature that, doesn't really do much else. And there's a good reason for why people have that prejudice, and it's that for most objects, such as faces, color is neither sufficient nor necessary for seeing objects, recognizing them, that is. So in this equiluminant rendering, it's very hard to even tell that there's a face there, let alone the identity. And luminance cues by themselves are perfectly sufficient, which is why most people who study vision don't bother studying color, because it saves you having to calibrate a display. So color can aid in things like face recognition under certain degraded conditions. As Yip and Sinha pointed out, you can kind of help segment the facial features if you have color, if the rest of the image is really crappy. But as they show, it's not color being diagnostic, it's not color for color's sake. You can get the same recognition benefit in a false color image. This recognition benefit is tiny, it's like 3 or 4%. If you know it's Lady Diana, to begin with, having the color doesn't really get you that much further. And I think it's for that reason, this very modest effect, that this isn't really what color is for. So what is color for? Well, I don't think we have a full understanding. It's probably not for one thing, but they don't talk about it. I think, though, this image gives you some clue. Don't you think Diana looks a little ill in the false colored picture? So color is a metric of health, and it's actually used in first aid. My sister is a registered nurse, and the complexion is one of the first things you were told to look at in order to assess health status, as any parent will tell you. So what is color for? Yeah, it certainly aids in scene segmentation and maybe in certain kinds of object recognition tasks, that sort of low-level image feature that I alluded to earlier. But it does a whole bunch of other stuff, ranging from grouping, attentional pop-out, categorization, memory. If I ask you what the color of a banana is, you can all tell me, even though you're not even looking at one. Health, assessing health status. Reward, there's lots of evidence that suggests color is actually a primary reward. And for nonverbal social communication. The colors we wear on our clothes, the jerseys we adopt for our teams, and so on. So it's for this reason that I think color is actually a really cool and understudied system, because it goes all the way from well-defined peripheral stimuli, a well-characterized retinal response, all the way up through to very fancy things that make us us. So my talk today has three parts which track roughly the feed-forward flow of visual information through the visual system. So in the first part, I'm going to talk about how color signals are encoded in primary visual cortex, V1, and a mid-tier area, V4. In the second part, I'm going to talk about color as a tool. It was a surprising discovery that we made, a tool for uncovering the functional organization, the organizational plan of inferior-temporal cortex. That's a big swath of tissue that's involved in high-level cognition. And in the final part of my talk, I'm going to return to some behavioral experiments that we were prompted to do by those neurobiological discoveries. So there's an interesting twist here, which is the neurobiology actually leading behavioral work. We often think of it going in the other direction. So as you all know, color processing begins in the retina with the absorption of light by three cone types. We often call them red, green, and blue, but that's a fundamental error. The peaks of the cones do not align with those primary colors. In fact, in order to see long wavelength light as red, you're only using the tail of the L cone sensitivity function. And I think this is a really important point, and I want to pause for a second and let it soak in. Primary colors, those color categories, they're the output of the brain. They aren't the input. And their compelling nature can lead us to think that there are things out there in the world, in the same way that Descartes thought vision was an active process reaching out from us, as opposed to a process of stuff coming in. And it's that basic question of how we derive meaning from those light signals, meaning in this primitive sense, like primary colors, but meaning in a more sophisticated sense, like you and I are on the same team, that I would like to ultimately understand. So the second surprise about color is that the triplet of cone activations in the retina doesn't tell you what color you're seeing. So we can define very precisely what that triplet of cone activations is. We know very precisely what the inputs are to the visual system in terms of color, but that doesn't tell you anything about what color you will see. And that's illustrated by phenomena observations like this. This is a nice image made by Bo Lotto, in which the blue and the yellow checks, indicated by the little circles, are physically identical when presented on a gray background. And I'm sure many of you have seen this demonstration before. So these contextual cues tell us, in this case, this is what the visual system is doing all the time. You wake up in the morning and you go to find your jacket in its particular color. This system is operating. This isn't like some weird lab demonstration. This is integral to how it is that we see color. And it's important because it enables color to become a property of objects, not a property of lighting conditions. In those original images, many of you might have noticed that the background illuminant was yellowish or bluish, but that isn't the thing that you choose to pay attention to, and in fact, your visual system very effortlessly discounts that bias. So in experiments I did a long time ago, I set out to understand the spatial and temporal contrast calculations that give rise to color constancy operations, and I did so by recording in primary visual cortex in V1. And at this stage in my career, it was very much, we know what the phenomenon is that we want to understand, and now I'm going to go to the brain and I'm going to understand what the mechanism is. And I have to say, my career has been marked by a radical shift from, now I don't even know what we're supposed to be studying, I need to do behavior to figure that out, and then I can go and look at the neurobiology. So in these experiments, which I won't dwell too heavily on, I recorded with microelectrode wires in macaque monkeys alert while they were watching a computer monitor on which I flashed little cone isolating spots. These cone isolating spots are analogous, they're directly equivalent to those Ishihara plates that you use for color blindness testing. They allow us to essentially tease out the way in which different cone types are modulating responses to look at what the inputs are of the cone types to single cells. And the responses of a single cell are shown here. So the background patch, that blue square, is a single patch of the retina or of the visual field. And on it, I'm showing with a color scale bar where in the visual field the cell responded, showed action potentials, when I presented either L cone isolating, M cone isolating, or S cone isolating spots. So they're all the same cell, and it's the same patch of retina. It's just the L cone input, the M cone input, and the S cone input. And you can summarize that receptive field with that schematic at the bottom. So what this cell really liked best was a red spot or an L cone increment on a green background on an M cone surround. And that characterization, that response has two properties. It's spatially opponent and chromatically opponent. It's so-called double opponent. These things are in textbooks. But at the time this work was done, there was very little direct evidence for them. So this provided sort of the first proof of double opponent cells. And they're interesting cells because you don't find cells like this in the retina or in the lateral geniculate nucleus. So these represent an important transformation of the retinal information as you go up from the retina into cortex. This is an operation that the brain has sort of chosen to make through selective wiring. What's interesting about these cells is that the calculation is relatively local. So these receptive fields are on the order of about 3/4 of a degree of visual angle. And that was surprising to me because we think of color constancy as incorporating information from across the whole scene. These receptive fields suggest that actually the calculation is much more local in visual space. So we can test that by going back to this original image. If the color induction or color contrast calculation that gives rise to the different percept in these different contexts is very local in space, we should be able to recover a stable percept by obliterating just that local interaction, and that turns out to be true. So I got very interested in this from my own background as a visual artist, trying to understand what are the strategies that artists use when they make paintings. Because as you can imagine, when you set out to make a painting, you start with a white canvas. These color contrast calculations mean that every mark that you apply to the canvas changes the color of every other mark. So how the hell does an artist ever make anything? And that's a struggle that I've had in my own work. You know, there's a little saying in art school that color is traumatic, because it's like, ah. Well, it turns out that some of my favorite, I did a deep dive in some of my favorite color painters. One of them is Matisse. And in his mature work, you often find these little white lines surrounding his colored marks. Now, we know that he was rich at this stage. He had lots of paint. He finished the painting. He signed it. These are finished works, okay? So why did he leave those little white lines? I think it's actually pretty straightforward. At the time, there was now an explosion of pigments available to him. So he didn't have to actually mix a lot of color. He could use it right out of the tube. And he wanted the color to look as pretty and beautiful in the finished picture as it did when he first squeezed it out of the tube. So in order to do that, he insulates the colored marks with these little white buffers. And he thought he was just doing something fun and beautiful. It was radical for the art community, and he was sort of an outcast in the initial stages of his career. And you can understand in a historical context why that's the case. At the time he made these pictures, the aspiration of a painting was to be a view into the world that left no trace of its own making. So you didn't want marks in the painted canvas. This is advertising. It's paint and canvas. It pulls back the whole curtain and says, look, I'm paint and canvas. That's like, ooh, you can't do that. Okay. Now, of course, the legacy of this in contemporary and modern art is everywhere, down to Mark Rothko and piles of pigment that you find Anish Kapoor making and so on. So you won't look at a Matisse painting again the same way. He did this like basically in every mature painting. Okay, so the predicted color tuning of the population of color cells turns out not to be uniform. It isn't a uniform representation of color. It's actually these color-tuned cells in V1 are very much biased. And for a while I was troubled by that, trying to figure out why it might be. But I think we can understand this in a Bayesian framework. So in a Bayesian framework, perception is a process of unconscious inference guided by these noisy sense data and an internal model of the world. Well, what's the internal model of the world? Optimality has argued, the theoreticians like Simoncelli and others have argued, that optimality requires a match between your internal model, that your internal model is somehow matched to the statistics of the world. Well, this makes a prediction that the statistics of the world should not be uniform. And that turns out to be true. These aren't my data. This is from Mike Webster's data looking at natural images. There's a very distinct bias in the gamut of natural images that manages to line up almost perfectly with the bias you see in these V1 color cells, which suggests that these V1 cells are actually encoding a prior about the daylight conditions. So about 10 years ago, I wrote a paper saying V1 encodes a prior about the daylight axis, and everybody was like, why is that relevant and interesting? Well, our work kind of collided with popular culture when this image showed up. So Adam Rogers at Wired phoned me up one late Thursday night and said, what's going on with this picture? And at the time, the picture hadn't really hit the internet big time. And I was like, I don't know what that picture is. And he said, well, what color do you see? And I said, well, it's kind of a brownish and a light blue. And he was like, that's not one of the choices. And so I asked him, well, what are the choices? And he said, well, people say it's either white and gold or blue and black. And you all know this story. And he said, we don't have an explanation. We've been talking to vision scientists and cognitive scientists all night. And they think it's got to do with variability in rods and cone ratios. And I was like, that's crazy. I mean, rods don't really work during daylight. They don't have anything to do with this. And we know there are massive variation in the cone ratios, the LM cone ratios, that have almost no impact on color perception. That just seems crazy. This seems much more likely to have to do with this prior about the daylight axis that's encoded in V1. And so the argument, which I think is now accepted, is that different people sort of favor one axis of that prior. So if you think of the light as being warm, you're going to discount the warm component and you'll see it as blue and black. If you think of the light as cool, you're going to discount the cool component and you're going to see it as white and gold. And we went on to show, and I won't bore you with the details, there's a very long paper in Journal of Vision where we sort of nail every aspect of this thing that you can think of. It's sort of like a mouse with a sledgehammer. But we go on to show that this thing is actually categorically perceived. So these are swatches of the color matches made, sorted by the report, the behavioral report. And we've shown quantitatively that these things form two distinct categories. So this isn't a unimodal distribution that's forced to be bimodal by virtue of an alternative force choice question. This is actually truly a bimodal image, and it's the first bimodally perceived color image that has ever been documented. So I think it's kind of interesting. We've gone on to show that you can train up a multinomial regressor on the color matches you make, and you can predict with almost perfect accuracy what color terms people will use. So this is, and this again is the first time anybody's shown that for color in any domain. It's actually quite striking. So the hypothesis is simply that if you disambiguate the lighting condition, you can force people to see it one way or another. So if you make the lighting clearly, warmly, you will discount it, and most people then see it as blue and black. And by the same token, you go the other direction, you see it as white and gold. And that's consistent with reports that people make, subjective reports people make about the lighting conditions. So to summarize so far, it seems that V1 is associated with a very early kind of operation that encodes a prior about the lighting conditions in a specialized population of cells, these V1 cells, these double opponent cells. But the role of these cells, as I said, it's speculation. It's still untested directly, and my lab is now going back to V1 to start to look at some of those calculations in more detail. But I think for me, as a graduate student and a postdoc, when I did this work, I was struck by how much extrastriate cortex there is, how much part of the macaque brain is used in vision that's outside of V1. It's like all of that tissue. I was like, maybe we're just looking at V1 because we happen to know where V1 is. It's like nicely demarcated, but it's not the answer to every problem. So I set about to try and understand how color signals are sort of tracked through extrastriate cortex. And at the time, there was really this dogma in the field that color is inextricably tied to object shape, If I ask you what the color of a banana is, you tell me the banana is yellow. Carl Gegenfurt was like, aha, there is evidence that color and shape are inextricably bound. And the argument went into V1 that said neurons are orientation tuned and they're color tuned. Color and shape are inextricably bound. And so the argument for extrastriate cortex was wherever you find shape tuning, you'll find color tuning. They're just mixed. The brain does some fancy, complicated thing that stitches these things together. They're just stuck together. And that just seemed kind of wrong to me. And I'll get to the end about why I've sort of pieced together why that was wrong. But the intuition was like, no, I don't think that's quite right. So to start to address that question, we adapted for fMRI alert monkey recording, where some students of mine at Olin College of Engineering designed a little plastic box in which the monkeys are trained to jump. They sit in a sphinx position, and they're trained to, it can go directly into a standard horizontal bore machine. So you don't need Nikos Logothetis' fancy vertical scanner, you know, because monkeys sit upright. You just tip the monkey this way and put him in the regular bore. It works just as fine. The monkey is trained to look at a screen, and you can show him whatever you like. You do a parallel human experiment. And I'll put a little note out there. We now have a dedicated 3T Prisma machine at the NIH for monkey imaging. And if any of you have an experiment that you've done in humans that's reliable and consistent and works, and you want to know if the same result is true in monkeys, get in touch with me. We have a large project doing a big comparison of monkeys and humans. So you can show any kinds of stimuli you like. In this experiment, I compared simply black and white and colored images. So these are equiluminant colored images. They're made equiluminant photometrically. And then I compare them with high contrast black and white. And I'm just going to pluck out 2 slices through that mid-tier stage. And you can see these are 2 independent data sets through 1 monkey. Right away, you can see some clear results. The first is that the color biased activation there is not in one big chunk. Okay? This isn't like V4 is the color area. And I think after Zecchi's initial studies, which were compelling and made their way into textbooks, everybody was left outside of neurophysiology with this notion that V4 is a color area, and everyone in neurophysiology was like, you find everything in V4 that you find anywhere else. It's no more a color area than V1. But this shows that actually there are these punctate little bits. They're about a couple millimeters in diameter. They're reproducible, they're bilateral, and they're distributed throughout posterior inferotemporal cortex of the V4 complex. So each of these little bits of brain comprises 30, 40, 100,000 neurons. We want to know what those cells are doing. So we use these maps to guide microelectrode recording experiments in the same subjects to control for variability and so on amongst the animals. And here is just an MRI of an electrode in place. And importantly in this work, I targeted not just the color globs, I call them, these color biased regions, but the adjacent bits of the V4 complex that are outside of those color globs. So a lot of people who had done fMRI guided microelectrode recording, I mean, these were some of the first work, were just going for the hot spots. And I was like, hang on a minute, we need a control. Like, maybe we should record outside the hot spots too. So we did that to compare how color signals are handled in these 2 little adjacent bits of brain. And this is a post-stimulus time histogram of a recording from 1 such cell. And I'll walk you through this recording for those of you that are not familiar with neurophysiology experiments. So on the x-axis is time, and on the y-axis is color of the stimulus. And the stimulus itself was a bar, optimally oriented to the cell's preferred configuration. And then on different trials, I changed randomly the color of the bar. The bar was on for about 200 milliseconds. And after the recording, I then bin all of the cells, all of the responses to a stimulus of the same color and rank them according to those colors. And you can see there are 3 kinds of colors. They're different luminance values, but they each, within each luminance value, they have the same sequence of color. So we can separate out hue from luminance. The top 2 rows show the response to black and white. Now, there are 4 pieces of evidence that suggest that this cell is coding color. The first is that very top bit, those white and black bars, these are like the dominant signal to the visual system is luminance. This cell doesn't care at all about black and white. It doesn't respond at all. Okay? Instead, it responds to equiluminant colors, and specifically, it shows some selectivity for those colors within that equiluminant set. And moreover, what I find super compelling about this cell is that color tuning is stable across changes in luminance. So instead of just showing that the cell doesn't respond to luminance, we're showing that not only does it not respond to luminance, but its color tuning is invariant to changes in luminance, which is kind of the first evidence that you have something that is like could code red independent of whether it's bright or dark. Now, we can capture this color tuning property in a polar plot like this, where color is plotted around the perimeter of the circle. The cell is then tuned to red, reddish-orange. And we found within the glob cells tuned to lots of different colors. Red cells, my favorite one is this one here, which is tuned to purple. And I like that one in particular because purple is an extra spectral color. There's no purple in the rainbow. It's what Stephen Fry calls a pigment of the imagination. So here you have a purple tuned cell. This is really a cell that is computing something out of its inputs. And that banded pattern you see in the PSDHs, that tells you that these responses are somewhat luminance invariant. The interglob cells on the other side, you can see, show a range of different properties, and depending on how you define color, you can extract a color signal from those cells, which I think is part of the debate. But I would say qualitatively, and it turns out quantitatively, these are different populations. They both, importantly, they both carry color information, but they clearly are using those color signals to do two different kinds of things. Okay, so in addition to that, we documented the color properties of the cells in adjacent cells and showed that neighboring cells show very similar color tuning. So these are two cells both tuned to blue. There's two cells tuned to green. And in long penetrations, we found the sequence regularity where the cells actually shifted their color tuning in a way that was consistent with the progression around a color circle. These are 7 cells recorded along a given trajectory. These data provide the first evidence for chromotopic maps, little microscopic maps of a color wheel, which is another kind of anatomical marker that this population of cells is actually specifically wired up to handle this operation. There's an exception there, which we can talk about at the end. And the scale of these color columns is about 200 microns, which of those of you who care about extra striate cortex is about the same scale as the motor columns in MT, the motion direction columns in MT. And this area is about, these cells are about the same stage in the visual processing hierarchy. We used multidimensional scaling of the color responses in a big paper we published in eNeuro last year to show that this population of glob cells, independent of luminance, actually captures very nicely a somewhat uniform representation of color space. And I want to back up for a minute and just underscore the point that this makes. We go from cone types in the retina that have biases to a representation here through V1 showing a bias of the natural scene statistics to a representation here that's basically uniform. It's providing a uniform gamut for the rest of the brain to then decide what it wants to use that gamut to do. And one of the questions we're going to ask is, how do we then get color categories? Where do they come from? You talk about red, green, blue, and yellow, and so on. Where do they come from? They must use this as the input. By the same token, the interglob cells represent luminance. You can see very nicely this carves it up into a luminance domain. So one of the questions we might ask is whether or not we can decode the color that you see just based on this population response. And here I don't want to get into the fancy details. The paper is published a couple of years ago. All we did was look at a population of 300 cells and then say, to a stimulus, which cell fired the most? And then I know the color tuning of that cell, and I simply say, I'm going to call the color of the stimulus the color tuning of whichever cell fired the most. A very simple winner-take-all with a little bit of stochastic noise thrown in. If the decoder works perfectly, you'd get a diagonal. And these are the results from decoding just 300 cells. You have almost perfect decoding. We can mind read what the monkey is seeing given that population. Why is that surprising? It's surprising because the dominant view in textbooks today is still that color is dependent on 2 channels, a red-green channel and a blue-yellow channel. And I hope that this is clear why that is so fundamentally wrong. Those color labels, first of all, are the outputs, not the inputs. And the channels that give rise to color are not these chromatic axes. They are a uniform representation that is then sampled by the rest of the brain to make it look like this is how you want to work. Okay, so to summarize part 1, the second part there, I showed that there's this encoding of color signals at the level of primary visual cortex in terms of the the prior of the natural scene statistics, and then we talked about how those signals then make their way up to a mid-tier to give you a uniform representation with these little microscopic chromatopic maps that could support color decoding. So I'll now turn to our work using color as a tool for uncovering the organization of infratemporal cortex. To start with, I just want to go back and look at retinotopy. So this is just how the brain carves up those meridian representations. So here is an inflated view of the macaque brain showing the representations for checker, the horizontal and vertical meridians. You can see that nice rotating or alternating sequence of horizontal and vertical meridian representations that carves up V1, V2, V3, and so on. But the most striking feature of these data is that infratemporal cortex shows almost nothing. Okay? These data provide, I think, the most, and it's not just our data, it's lots of data like these, provide the most compelling evidence that there is a disjunction, a transition between about V4, between a representation that is constrained by the retina to a representation that adheres to some other kinds of rules. And I don't know what that transition is, and we have very little idea about whether the organizational principles in the back part of the brain apply to the front part of the brain. So to identify that, I think there's been these two kind of big models, which I'm not going to get bogged down in, but you have people like Nancy Canwisher who say you've got a blueberry-sized chunk of brain that's responsible for faces, and it's like there's the parts list, and we have to figure out what all those bits are. Very rigorous quantitative characterizations. And then you've got people like Jim Haxby who say, well, it's kind of a gamish of lots of different stuff, and everything's participating to help out, and it's like this randomy thing, but that randomy thing has information in it, and we can decode it, and it's like neither of these things seems particularly biologically plausible. Okay, so we replicated the same experiments. Sorry, is there a disagreement? Nancy is a good friend of mine, and I've never met Jim, so yeah, maybe they don't talk like that. Okay, so here what we've done is calibrated the stimuli so that they cause the same level of activation, the color stimuli and the black and white stimuli, they cause the same level of activation in V1. So we're actually using V1 to titrate the relative contrast of our color stimuli and our black and white stimuli. So we show black and white gratings and color gratings, and you see nothing in V1, and everyone's like, but V1 should be responding lots of it. They do. V1 responds tons to both of these stimuli. It's just balanced by design to elicit, to allow us to pull out the color-biased activation. Okay, so you see the little globs in V4, posterior IT. You interestingly see the stripe pattern in V2, a little evidence of the stripe pattern, cytochrome oxidase stripes. You see a luminance bias in V3, which nobody talks about. Like V3 doesn't do a lot with color. What's happening in IT? Well, there we were surprised to discover these giant globs of tissue. Okay, and they're somewhat systematic down the length of IT, and I was totally surprised when we uncovered these results. They were surprising in part because IT is like high-level object vision, thing one, and thing two, we already have, by the time you get to V4, a complete representation of color. Why the hell do you need big chunks of brain in IT doing color? And there's as much tissue that's color-biased in IT as there is face processing tissue. And nobody studies it. It's like, but this is a huge chunk of brain. What the hell is it doing? It made me, like I remember the conversation with Josh Tenenbaum. He was like, that can't be true. We'll have to rethink what color is for if that's true. And I was like, well, it turns out true. Let's rethink what color is for. Okay, so we use the face patch system as a functional marker to understand this organization in IT and discovered that the face patches and the color bias regions are systematically related to each other, almost like these kind of like beads, parallel beads down IT. This suggests that there's some fundamental organizing principle here that's giving rise to it. Well, it turns out anatomical work agnostic to function has identified 4 nodes down the length of IT. This is from this big review paper from Ed Kravitz and others at the NIH. If we just take a course level registration of this anatomy and you put down those things, these anatomical nodes predict the location of those functional domains. It's kind of a beautiful form-function relationship that suggests that those nodes are actually related or giving rise to that functional organization. More deeply, I think it tells us that whatever the rules are that govern the information flow down the face patch network it's the same analogous set of rules that's governing the interaction down in these parallel tracks through IT. And that's powerful because it means instead of the face patch system being some weird anomaly, little islands of dedicated tissue in a sea of undifferentiated soup, we can think of the face patch system as one manifestation of a canonical set of rules. And now the question is, how does that canonical set of rules get implemented in these different streams and for what purpose? So you can think of these things as essentially doing the same operation, just with different inputs and functionally connected to different outputs. So they derive their functionality by virtue of that functional connectivity, which I think is an argument Alfonso and others have made that I find very compelling. So these data also result in long-standing controversy, which is why it is that people who suffer from acquired prosopagnosia often have achromatopsia, and it's now been shown that's also symmetric. And it's probably because these bits of brain are sitting next to each other, and a stroke is agnostic to, the functional domains. So there are a number of questions this raises. The first is this organizational structure similar in humans? I had a little, point of pride that this was an experiment done in monkeys that then led a functional experiment in humans. Usually it goes the other way around. So we then went back to go and look at humans, whether or not this was true. And I teamed up with Nancy Canwisher, who knows something about human fMRI. It turns out it's a lot easier than monkey fMRI. But in any event, it turns out that you see the same basic structure in humans with these color bias domains sitting wedged between face patches and place patches. This shows the ventral view because we wanted to understand what the relationship was between these color bias domains and shape processing domains. So remember that question that I started with about color being inextricably tied to object shape? Well, lots of people have shown that you can show scrambled objects and intact objects, and you pull out these big chunk of brain, LO and PFS shown here on the surface. So what's the relationship of those shape processing domains to these color bias regions? And it turns out they're almost entirely separable, except for that very anterior bit. And we can quantify it here in these bar plots. O stands for object, and SC stands for scrambled object, and the cyan bars are the colored versions, and the black and white bars are the black and white versions. And you can see just right away, those color bias domains are showing a stronger response to the colored bar, the cyan compared to the black-white, but they don't show a difference between the object and the scrambled. If anything, there's a slight bias for the scrambled, okay? Probably because it's introducing more color boundaries. Conversely, those anterior bits are now showing a bias that's mixing both color and form. So this provides evidence that at the level of sort of extrastriate cortex, you have a functional dissociation between color processing domains and shape processing domains, which we've quantified with shape bias and color bias domains, and you can read about that in the paper. So the second question is, what's this basic organizing principle? Well, I was really taken with these studies when they first came out from Malik's group, showing these coarse eccentricity biases throughout IT. And it prompted us to go back and look in our same monkeys what the eccentricity representations were. And to cut a long story short, what we find is that there are, in fact, eccentricity biases down the length of IT, even though we don't see clear retinotopic mapping. And those eccentricity biases, shown in yellow on the right-hand plot there, with the asterisks indicating the fovea, those eccentricity representations actually predict the location of these functional domains. So it looks like what you have is this repeated series of eccentricity templates down the length of IT. And where does that come from? I love this idea. It's totally speculation from John Ullman, Ullman and Caos, where they said that a common mechanism of evolution is replication of an ancestral domain. So in the case of photoreceptors, we duplicated the L cone to get an L cone and an M cone. In the case of the body plan, we duplicated the vertebra to get arms and legs. I mean, we see this over and over again. So I wondered whether maybe what we see here is evidence of that evolution playing out, where we've duplicated that ancestral retinotopic area, which was the argument Ullman and Caos used to account for the expansion of V1, V2, and V3 and their shared meridian representations. We just carried that mechanism all the way through down the length of IT. And that then predicts that there will be a bias of the central representation, visual field representation in IT for faces, and a peripheral bias for places, and an intermediate bias for color, these colored bias domains. And that turns out to be true. And it raises this kind of quirky question, which is like, in a cognitive space, what's the scale of color? And I think the scale of color is the sort of stuff you can grasp and hold, which then makes some clear predictions about the nature of color use, what we use color for, which I'll get into the last bit. So just to summarize the second part, we've used color now as a tool to uncover what I think is a fundamental organizing principle in inferotemporal cortex, and shown that that principle is predicted by anatomy and is consistent between humans and macaque monkeys. So it suggests that maybe our ancestral, common denominator evolutionarily had this. So what is this bit of brain doing? I have no idea. This is my current research program. Here's just some wild speculation. These things map on somehow to those things. I don't know, right? But we've got lots of stuff to work with. Okay, so in the last part of my talk, I just want to touch upon some recent experiments aimed at understanding what color is for. So to gain traction on this problem, we first set out to just look at the color statistics of the parts of the world that we choose to label. So remember I talked about the cognitive scale of color being the size of objects? Well, what are the colors of objects? It turns out, strangely enough, we know a lot about the color statistics of natural scenes, and we know nothing about the color statistics of objects. And for a lot of people, when I raise this question, they're like, that's crazy. They'll be like every color you can imagine, because we can arbitrarily color the world, and so we'll make objects any color. That just turns out not to be true. So we analyzed the color statistics of objects from a large database of images, and this is just 10,000 randomly drawn pixels from objects and from backgrounds. You can see right away there's a statistical bias. And in fact, that statistical bias is systematic, such that object colors vary in their LM ratio, and animate and inanimate colors vary in their S-cone ratio. So color information we showed with a machine learning kind of hog algorithm. The average color in an object tells you as much information about its identity as its shape. Okay? That's crazy. Why don't we use color in these machine learning algorithms? I talked to Tommy Poggio about this, and he was like, color isn't useful for Hmax. We got rid of it. I was like, but it's as informative as anything else. Like, it's crazy. So we went on to look at you can, in fact, the famous Krieger-Skorda data set, you can decode the animate and inanimate identity with the average color beautifully with a linear regressor just looking at the average color in the pictures. Okay, so that then returns us back to IT. There, we just looked at the color tuning of voxels and correlated it with the color tuning of objects, and you see this beautiful organization down IT, where the color information, the color tuning, predicts that animacy, inanimacy organizational structure that you find down IT. And it's there in IT, and it's not in V1. Okay? So this raised a question for me about, like, how do people use color terms? Returning to that initial question. So when I first joined MIT, I went to Ted Gibson, who's Canadian, and we had a beer, and he's like, fun. I'm Canadian now. And he was like, so if you could do any experiment in the Amazon jungle with a population that has no exposure to Western objects or very little industrialization, what would you do? And I was like, oh, I'd love to do a color naming experiment that wasn't confounded in the way of the famous Berlin K experiments, which are confounded by experimental bias. Berlin and K went in and said, you're only allowed to use monolexical words. It has to be in your own language. They can't be borrowed from anything else. They can't be associated with a specific object. By the time they got through teaching everybody what they were allowed to say and not say, people gave them exactly what they told them to give them, which were the English color terms. I mean, it's like the craziest bit of tautology in science. So I said, let's just let people say whatever they want to say. So he was like, that's a good idea. So 2 weeks later, he wrote back and he said, can you send me all of the materials we need? And I was like, are you serious? He said, yeah, I'm going down next week. So we got a car battery powered light box and color chips, and we set it all up, and we collected tons and tons of data. It's something I really love about TED. We're on the same page. You need lots of data. So we collected lots of data in a complete free-form experiment where all we said was, just tell us what color, just tell us how you would describe this to someone else, these little color chips. And there's Ted collecting the data. So you can think of this analysis as a kind of color game where I've got a complete color array in front of me, you've got the same array in front of you, I pick a chip, I use a word to describe that chip. How many guesses does it take you to figure out which chip I'm talking about? Okay? How surprising is the color term? And for those of you that like math, there's some math. So this just shows For all languages in the World Color Survey, we did it first for the Chimani to validate it, and it turns out we can then use the World Color Survey data. This just shows the average surprisal of 80 chips across the array, rank ordered according to surprisal, for languages rank ordered by fancy color names to crappy color names. Okay, so these languages down here, these are like your primitive, I put it in quotes because I think it's BS, but they're languages that just don't have very good color words. What Berlin and Kay would say, evolutionary, very old, stage 1. There's fancy English and Spanish and Italian right at the top. We're like really good, we got great. The structure that you see in terms of communication efficiency is universal. We all communicate warm colors better than cool colors. And that doesn't matter how fancy your color naming is. So what underlies this universal structure? Well, it turns out it's the color statistics of objects. It's the stuff we choose to label in the world. So there's a very nice correlation between the average surprisal and the probability that that thing will be an object color. Now you might be saying, yeah, but we live in an artificial world. We can color our objects however we like. That's true. But if you're setting out to color an object, you're going to lean on evidence that you have from the natural world. It's like if you want to start an Indian food restaurant, you put it next to another Indian food restaurant in town because everybody knows to go to that one to go to Indian food restaurants, right? So we're going to borrow those statistics, and that turns out to be true in color. So there is more structure in this, and we have a paper under review now. Who knows if it'll get published? It's one of those year-long battles. Just looking at the structure within this graph. So here you can see there are some interesting transitions. So purple actually breaks out of these low communication colors and emerges as a high communication color relatively late. And we've quantified that in the graph on the right. And that's really cool because purple is a synthetic pigment. Most purple in our environments comes about synthetically. And so this tells us that when you make purple, when you make a color useful, as you do by making it synthetic, you promote its usefulness, and now we boost up its communication efficiency. We actually have something to talk about that's relevant for talking about. So we did this. Do I still have a few minutes? Negative 5 minutes?
You have one more minute, but I'm more than happy to let you continue.
Are you more than happy?
But how much more do you need?
I need 5 more minutes.
I'm going to give you 5 minutes.
Okay. I mean, I'm happy to shut up and just take questions. All right. So we did this experiment in the Chimani trying to figure out how people use color terms. So we did this contrastive labeling task where we presented people with this thing and said, what is it? And then we presented a few seconds later, what is that? Okay. Now it turns out it doesn't matter if you do it in parallel or in series. People say pepper, and then in English they say, red pepper. It's like, oh, you want me to talk about the color ICI. So it's a way of implicitly eliciting color terms. So there's the Chimani. You do the first one and the second one, they never use color terms. Like, it doesn't matter how many times you show them the same object for a natural object, because they have a specific term for that thing. They have a specific term for the ripe pepper and the unripe one, or the red one and the unripe one. In English, we use color terms. We cotton on to the game. What happens when you use artificial objects that are objects they're somewhat familiar with? Now, all of a sudden, you get that priming in the Chimani. So the second presentation of an artificial object elicits a color term. Why? Because by making it artificial, you have promoted the usefulness of color. You've isolated the only way in which you can tell that thing from something else. Now all of a sudden color is useful. The Chimani are perfectly happy to use a color word. So we're tracking this population over time to see, as industrialization moves in, what happens to their color naming. So the last little second thing I'm going to talk about is this, returning to my banana example. So what is this? It's a banana. What color is it? It's yellow. Well, it turns out bananas can be lots of different colors. In my house, they start out as, green, and then they're yellow for a hot second, and then they're black, and we throw them out, right? In fact, statistically, most of the bananas you've seen in your life are probably not yellow. So it's kind of remarkable that you tell me that bananas are yellow, right? Why do you tell me they're yellow? Because that's the state of the banana that you care about. So color is telling you something not about identity, it's telling you something about valence, about whether or not you care about it. And I think that is a completely independent parallel operation that we have really thought very little about. The identity of the object communicated by its shape does not change much throughout the life of its shape, throughout the life of the object. The color is the transition that tells you quite a bit. And that turns out to be true for humans. So when we relate to each other, certain parts of my face change, and one of the dominant things that changes is complexion, color. I communicate all sorts of really important stuff by my color changes, health status, emotion, and so on. Okay, so to try and tease apart the relative contribution of color to object and social perception, we did a very stupid experiment. We asked people to color label, color match objects in a low pressure sodium room. So these rooms, this old monochromatic light, they completely get rid of color vision. It's a kind of experimental paradigm borrowed from genetics. If you want to know what the function of gene X does, get rid of it and see what happens. We want to know what color does, get rid of color, but leave everything else intact and see what happens. So we have people color match real world objects. So we had African American actors and Caucasian actors come in, and we color match bits of their face, and we had objects, strawberries and so on, we color match them. And it turns out there's a set of, these are real world objects. So there's like people sitting there and you're like, it's a little awkward, and you're like, what's this? And you get over it. These are the color matches people make under normal white light, which was done after the low pressure sodium as a control. So the dark bands are the African Americans and the light bands are the Caucasian. You see this race-based effect, which is a cognitive penetrance. If you know someone's race and they're African American, you're more likely to say they're darker skinned, and in fact we recover that. Tomatoes and strawberries and everything else, sort of ping pong balls are exactly what you expect. What happens under low pressure sodium light? Here are the results. Most of the objects are in the orange-brown, yellow-brown, consistent with the color of the light, except faces. And particularly, this just shows the same match to forehead with and without a mask. So it's exactly the same thing in the subject. We just put a mask in front of their face and then a little. So this color bias that you see for matching faces under these conditions isn't about like the complexion or the vasculature of the stuff. This is about a contextual effect, made implicit by the shape context. Okay? And it's really incredibly robust. 100% of subjects experience this. People come out saying their faces look green, they look sick. So this is a very strong memory modulation, shape modulation of modulation by memory of face color appearance. And we've analyzed the color statistics of those matches that people make and shown that this color bias is actually selective for the LM axis, the chromatic axis we achieved with trichromacy. And I think that's really cool because there's lots of dimensions of chromaticity in the face that activate all the cone types. This shows that the bit that you encode in memory is about the LM dimension, the dimension that you got as a result of trichromacy, trichromatic evolution. And what is communicated by the LM dimension? The dynamic stuff that we care about, like whether or not you're angry or mad at me or healthy or sick and so on. So it's kind of very compelling evidence that shows that at some fundamental level, why do we have color? It's got to do with that thing that I started with. A cool kind of test of this idea of the importance of face color in color constancy was provided by Rosa Leifer-Sousa in my lab, who said, well, if that's the case, we should be able to influence the color judgments people make of the dress just by tinting the color of the model. And that turns out to be true. So we've now gone on last second to measure the color tuning properties of face cells, because everyone has assumed that face cells should only respond to black and white because black and white is all you need to recognize faces. Turns out, yes, black and white drives face cells the best, but they show a subtle color tuning, and that color tuning is consistent with a prior about the likely colors of faces. And with that, I thank you for your attention, and I'm happy to take questions.
All right, you know the drill, so let's go.
Mel. Oh, no, I know the drill. No, I don't know the drill.
Well,
Sorry,
this is
I should have learned from Sharon yesterday.
This is my first question at the conference, so I feel. Yeah, absolutely. Yeah.
Mel tweets Mel's first question.
Yeah. So, I mean, that was a wonderful talk, and you've made it abundantly clear that color is an important property of the surface of objects that helps you understand and can be contextualized by other things in the array. What you didn't talk about, and I'm sure you've been thinking about, is the other features of the surface, such as glossiness and texture and transparency. And I wondered, have you begun to explore how they may be related or interdigitated with the representations that you've shown for color along the temporal lobe?
Yeah, so thank you for the question. And you'll notice, I hope you notice, that I'm very careful when I use domains in IT. I call them color-biased tissue, which is like cumbersome and annoying. Other people call them color patches. You know, we discovered them and they get to rename them. I don't know how that works. I don't like color patches, and I don't like color patches for exactly the reason you have honed in on. We have no idea what the operations these cells are doing, and it seems very unlikely to me that they're doing just something related to hue. In fact, I think they're doing something related to all of the kinds of things that you use, all of the kinds of information that you use to assign value to something, or at least to assign object status to something. You know, the texture of stuff, wood. I mean, you're a better expert than I am on it. I mean, I'm new to this game, but I'm very sensitive to the fact that we can't overreach here. We've used color as a tool to try and understand that architecture. The color bias we find is very tiny. I mean, you have to titrate it in order to get color activation balanced in V1 to pull it out. But once you've done that, it's highly reliable and consistent. You see it in every monkey and every human. What are those bits of brain doing? You know, I'm very keen. I mean, this is one of the reasons why I tracked down Kentridge's stimuli, because he's got very nice stimuli manipulating, and you guys have some nice stuff too, actually, manipulating the textures of objects. You know, there's lovely work from Qasim Zaidi's group showing that if you just have people go through and identify what the texture is of something, you can actually pull out with a very simple equation the Fourier component, the frequency component, that drives a response of puffiness or glossiness or wetness or solidness. I mean, it actually turns out to be a very straightforward, simple kind of operation, which seems like something we could implement. Whether or not it's implemented here, I don't know, but it's a very nice place to start. And we're gearing up to do some fMRI experiments to see, are these domains sensitive to those image features?
Well, thanks.
Beautiful. I must have missed it. So you have these repetitions all the way down. What do they do?
What do they do? Yes, that's the question. We don't know. So I think the best evidence to date comes really from the neurological literature. So, you know, when I first sort of made those observations, I was like, what do they do? You know, I sketched out this idea that maybe you have a representation of uniform color in posterior IT that's then made accessible to these later stages. Maybe the intermediate ones are doing something related to categorization, which is analogous to the kind of contextual, global contextual operation that gives you a face from image features. And the later stuff is doing something that's more specifically tying memory or a label, either in a human's language or in monkeys, a kind of behavioral relevance to that thing. Okay, that seems speculative, and maybe it's where we're at in the field, and it's a good place to start. But there's beautiful work, actually, you know, that you will know and you contributed to, showing that you can have selective deficits of posterior bits of brain that leave you impaired in a kind of achromatic state, but don't impair your ability to know what color an object should be. And conversely, more surprisingly, actually, you can get the opposite kind of lesion where you're perfectly capable of telling me what color it is, but if you ask me what color a pig is, it's like, I don't know. So there are these, and there's the lexical component where you can know what color it is, you can match it to an object with an image, but you can't tell me the words that you should use to map onto it. Now, I'm characterizing that literature as an outsider, where I've got like the distilled abstract that's very clean. It's like, you know, it's like, well, it's a little bit better in them and a little worse, and it's, you know. But I think the evidence is there that says, hey, this is consistent with this idea that you could dissociate these different high-level operations. embedded in your question and in what I've talked about is the role of experience. So I very much am pushing against the dominant trend in visual neuroscience, treating vision as a feed-forward operation where you have a kind of Jim DiCarlo thing where you got a monkey and you're showing him a rocket ship on a background of a jungle. And lo and behold, you find a cell that responds to those image features. Yes, there's a feed-forward component that's driven by these shaped features, but ultimately, what makes the brain powerful is its ability to actually integrate that with your demands, with what you're placing on it. And I think that's where thinking about the outputs of these bits of brain, what they're functionally connected to, is going to tell us, we can reverse engineer what are the operations that these things are actually geared up to perform with the inputs. So, there's evidence that says that the more ventral bit in IT anterior goes more to the hippocampus, the dorsaler bit goes more to frontal cortex and the amygdala. It's like, what do those asymmetries tell us about the operations within them? And, I'll pull back. The brain is a complicated thing. These are billions of cells. I'm not going to pretend that I know the answer. But I think we're at a stage where speculation is probably all we can do, and maybe we can be creative about that speculation. So I welcome yours.
Hey, thank you for that. My question relates to your last point, actually. So you talked about, and I'm going to get my terminology wrong, I'm sorry, double opponent cells and how they reflect the statistics of the scenes in the natural environment. Is that something evolutionarily that they reflect, or does that change as our experience changes?
Yeah, I don't know the answer to that question. We really don't know. Right now, what we're doing is a series of experiments in monkeys where we control the statistics of their visual diet to try and get at some of those questions. But I would point to some of the developmental work that suggests that V1 is wired up largely in utero by way of retinal waves and so on. And if I make an extension that you have orientation-tuned cells wired up at birth without natural scene statistics, I might argue that other properties of V1, like the double opponent bias for natural scene statistics, is similarly wired up in development by virtue of the particular patterns of connections between, in this case, it's really S-cones driving through on bipolar cells. That circuitry is established very early in development. And I think it's a selective tapping into that circuitry that V1 gets, which probably is related, is driven by timing of those waves and so on. It's there's a bunch of neurobiology in there, but the answer is, I think it's probably innate. But I don't know.
This is a bit of a fuzzy question, but I was just...
It's early.
It's early, right? So in the domain of form, okay, I think it's reasonably well established, for instance, by Mel and many other people, that that visual information about object structure can interface with motor systems quite independently of its use for object recognition. Mm-hmm. Is there anything for color in that sense? Because everything you showed about the derivation of color was all cortical, and as far as I could tell, in the geniculate striate pathway. Mm-hmm. Is there anything that you know of where...
So when I review those papers, I often say, hey, have you ever thought about color? And the authors say, no, it's irrelevant. And I say, yeah, I'll get back in my box. So, look, if color is this important a feature just statistically for identifying what an object is...
Well, that's why I'm asking.
Yeah, that's why I asked the question, but I haven't studied parietal cortex. I don't know. I have no idea.
Hey, thank you for the talk. It was great. I've done some studies with fruits and vegetables as a localizer kind of thing, and surprisingly, the areas that I get when I contrast fruits and vegetables with a bunch of other, and these are grayscale images, so there's no color there. The areas that I get when I contrast fruits and vegetables with a bunch of other categories are between PPA and FFA in the fusiform gyrus. So. And there's no color in the picture. So does this help with some of the functional questions?
Yeah, I think it's probably related to Mel's question. There are also these other features that co-occur with color that you're using to parse up the valence or importance of that object that have to do with texture and so on. And I think that's probably what's activating that middle bit. I mean, the color bias that we discovered for the statistics of objects, there's a funny backstory, which is, like, I thought that all objects would, I mean, if you collected all objects in the world, you would see a uniform representation of color space to the extent that we all have some coarse agreement about, you know, what constitutes a uniform color space. So I gave the lab like a couple $100 and said, I want you to go out and buy objects of different colors to span all color space. Okay, they came back, and I was like, natural objects. They came back, we did spectral readings. All of the natural objects are in one quadrant in color space. Even blueberries turn out not to be blue. I was like, try harder. And so we it just turns out there's this very striking bias. So, you know, there's also a bias in terms of the texture of those things, and their translucency, and their glossiness, and their hardness, and lots of properties.
Fascinating talk, Bevil, thank you. I was wondering, so I was kind of surprised, well, maybe just because I don't know very much about color processing, but at the level of V4, I think you showed that you can decode basically the color just by taking a winner-takes-all model, right? Just one neuron that encodes fires the strongest. So I was wondering, does this mean that at this level at least, color doesn't rely on population coding, so you just have single units representing certain color?
That's the question we're currently testing with microstimulation experiments in the context of well-defined behaviors. But yeah, I think that is the crazy, surprising prediction that this work makes.
I just wanted to pick up on the interchange between you and Brad about action and color. I think color plays an incredibly important role in action, and I think it plays a role
Take notes, Brad.
Where there are a lot of opportunities for learned association, so that if you're going to pick up an object, what you want to do is to know how much force to apply, where to actually put your hand. And so things like the function of the object, its fragility, its compliance, its temperature, all of those things will be signaled through learned associations between the surface properties as well as the shape of objects and your interaction with them on past occasions. And so color plays an incredibly important role, but I still think it's largely a ventral contribution, not so much a parietal contribution, where I think you can, from first principles, derive things like the kind of overall shape and size and orientation of an object from optical physics. You don't require color as a contribution.
And those shape features are the ones that are most sort of principally important for grasping.
For the initial part of the grasp, absolutely. Yeah. For the final manipulation and the forces you apply, I think that is where things like color become incredibly engaged.
And yes, although it's probably very coarse, like if it's animate, you want to be gentle.
No, absolutely.
It's probably pretty...
All of those visual features that are processed in the ventral stream become relevant.
Yeah.
More questions in the back there?
Shall I give it to him? All right.
He'll speak with his back.
These are just two silly questions. The first one, if you have anything to say about synesthesia, why is it that it is particular kinds of shapes, visual forms, that are most likely to be associated with synesthesia? Okay, why not faces, for example? And the second question is, have you looked at the connectivity patterns among these patches, these various patches that you're identifying? You don't want to call them patches, I forget what to call them.
Color bias regions. That's fine. It's fine. Not silly questions at all. So the synesthesia question, the only reason I chuckled is that I was surprised that hadn't come up earlier. So, you know, I'm not going to answer your question directly because I can't, but what I would do is I'd take a step back and I'd say, hang on a minute, what is color for? Well, I would say color is a trainable system that it's important it's trainable. And it's trainable, why? To detect things of likely behavioral relevance. So there's, I think, maybe a principled hypothesis, you know, for the first time, just put it on the ground, like there it is. Color is a trainable system used to identify objects of likely behavioral relevance. In that context, it's unlike lots of other visual features. Motion doesn't do that, shape doesn't do that, and so on. So then what is synesthesia? It's this thing that comes about because of experience, typically experience yoking shapes and colors. So I think some of the most compelling data for synesthesia shows that the color shape associations are predicted in a large surprising number of synesthetes by the colors of fridge magnets, right, which were at a developmental stage and their experience really critically important and so on. I think it's crazy how many people have synesthesia. If I ask this room, like, you don't have synesthesia, fine, but just play my game, what's the color you associate with the number 0? Most people say black or white. Black, white, or gray. If I say, what do you associate with 1? They say the opposite one of the one that they just said. I mean, it turns out it's like, these are like there are real data out there. It's like, it's a significant trend in the population. So there are ways in which we have mapped, or we map that binary classification of black and white onto a binary classification, and it's probably predicted by how facile you are with computers, how much computer use you use. You're like, do you use binary? Then you're probably more likely to have that synesthesia. So it's like it's particularly sticky. Color is the thing that's particularly sticky.
I was especially interested in these results where you showed us the pop-out effect, and you get pop-out effects when I give you black and white letters or digits.
Yeah.
And so going back to your argument about priors and how this perception of color may come about, I mean, there are no relevant priors in black and white digits that will give you to bias you to say 1 or 2. Now, you say, well, it is something you've learned, but the response seems to be a pretty, quote-unquote, low-level effect, if that's how you get the pop-out effect.
So that's what I want to push against. I think, the appeal to our intuition about how these phenomena like pop-out feel to us is misguided. And I have great respect for you, so I hope this doesn't go the wrong way. But I think the important piece here is that you can experience it as seeming to be immediate belies the extensive training and neural operations required in order to support that behavior. And we know that developmentally. So kids don't experience, it takes them a long time to learn color labels and to abstract color away from shape and so on. So I think, I'm very curious to look at pop-out in infants. And I don't know that infant literature, someone must have done it, but I'd be curious to know, like, is this something that you had to really teach them to get to do? Is it actually innately there? I'm agnostic about that. But I think, that it might seem immediate is not compelling enough for me to say, oh, this is low level, because then I go down the low level stimulus feature argument.
Well,
and yes,
I agree with your reasoning, except that the kind of evidence that perception scientists, I'm not one of those, brings to bear on the question is not merely only the subjective experience, but it's a fact that if you look at the response times, there are particular signatures that have been interpreted as showing that something is not dependent on whatever.
And that's
so that I think is
So here's maybe if I say anything important today, here's what it would be. Those data that point to it being really early, the connection between those early signatures and it actually being early as in implemented in V1, there's no data for that. We find latencies in IT that are as early as latencies in V1 for neural recording. So it's like, this is not like the notion that we have that vision works by a baton being passed from the retina through the LGN all the way up where, ta-da, I'm a color, is like, it's kooky. And with all due respect, I have that same cartoon in my head. It's much more like, I call it making soup. You've got a dynamical network that's busy doing stuff, and then you try and perturb it and push it in different directions. And if it's really trained to care a lot about color, it's going to be perturbed quickly. But that doesn't mean that it's an early operation. It could be driven by frontal cortex. You know, Bob Desimone showed very nicely these connections between frontal cortex around that little piece of lateral prefrontal cortex connecting right down to mid-tier V4, which suggests that you've got a modulation that he's like shaking his head. All right, someone's live tweeting this.
I think you're misconstruing my early late. My early late doesn't correspond to neuroatomic sites. That is the furthest thing from my mind. Early late for me is understood as level of processing, and I have no idea where, how in the brain these things are done. But in terms of you were able to order a series of computational steps that your theory claims to support, then it would be at the early stages of that computational sequence. So to rephrase the question then, or to have you try and answer it, is something that is occurring presumably at a level prior to more cognitive operations. That, I think, is the way in which people in this field have interpreted those kinds of results, not V1, V2, V3.
And that is why this exchange is so important, because people in neurophysiology hear cognitive people say early, and they think you mean V1. And sometimes it gets explicit, you know, those papers about whether consciousness happens in V1, that whole little misguided dead end in neuroscience, like that's kooky, right? For exactly that reason. Now there's a separate question, which is like mind and brain come together. It's like, well, can we ever try and understand the way in which you use early in neurophysiological, neuroanatomical terms? Or is that just an enterprise that's fraught and misguided, like trying to understand flight by looking at a bird's feather? I mean, it's like, that's not going to be the right way to even think about it. And I'm more on your side on that way. Like, I think ultimately it will be problematic.
Anybody else want to jump in on this?
I have one more thing answer to Philip's question. Am I allowed to jump in
Sure.
myself?
Make it quick.
Okay, I'll make it quick. So if it is like winner take all as I've described, you might come back to me and say, okay, if I'm God and evolution or whatever your construct is for making me, why didn't I just endow the retina with detectors for each of those colors that I care about? Okay? And the answer there is some species do that. So the mantis shrimp does that with its like millions of detectors, and behaviorally it has no better color vision than you do. So why do we do it the way we do it and they do it the way they do it? I think the answer has to do with the constraints imposed on us by having a simple eye that requires we use that eye to do lots of things, one of which is bring an image into focus, which has to then wrestle with chromatic aberration. So because of that, we end up having to do all of this pre-lifting to get us a representation at about, mid-tier that can then be used to do the things that a mantis shrimp uses color for and every other species uses color for.
All right, I just want to make a quick comment. I have no question. In addition to recognizing what an incredible talk that was, I hope everybody also was very aware how incredibly cautious Bevil is in terms of speculating. There are people in this room who, if these were their data, would be building, I'm looking at Alfonso actually, sandcastles that would likely crash, but would be very interesting. So it is incredibly refreshing. I've heard this question about function we've been asking him from the get-go, and that was the most complete answer, the one you gave Alfonso. Bevil is very aware of the double dissociation between achromatopsia and color agnosia, which he described. And it seemed to me one hint up there, if I read that correctly, is that in the more anterior regions there is an overlap between object form and color, which could start you to weave a story about color agnosia.
Absolutely.
So let's all, we'll talk about that during the break. Sharon, what time do they come back? Well, it says 11:15, because shouldn't we give them a full hour for coffee?
Well, we're going to cut into lunch then.
No, we don't want to cut anything. 11:15, okay, 11:15 for Morgan, please. And please, let's thank Bevil one more time.
Yeah, well, they'll